This is the first podcast of 2024, and today I have an expert on frontotemporal dementia. And many of you may have heard of this recently. It's been in the news because the diehard guy, Bruce Willis, has been diagnosed with this disorder, and it's kind of been in the news and how his wife is dealing with it and how difficult a disorder it is. Professor Seeley, he's a Zander Family Endowed Professor in Neurology at the University of California, San Francisco, where he's also on the faculty in the Neuroscience Program and Institute for Computational Health Sciences. Um, he's a neurologist, a neuropathologist. And could you start, Bill, by talking about your early life? What got you interested in the brain? And did you have parents that were physicians or in the sciences?
Yeah. I didn't have parents who were in the sciences. I had grandparents, great grandparents, and as it turns out, great great grandparents who were in medicine and science. And I didn't even know all of that when I was kind of growing up and trying to decide what to do with my life. I just made a promise to myself when I was in high school and early college that I was gonna spend my time on whatever I thought was most interesting. And I hope that that would be something where I could also make a contribution to society and in a positive way. Um, but I didn't know how it was all going to work out. And in fact, when I was an elementary school student, I remember being asked what I wanted to do when I grew up. And I said, the last thing I want to do is be a doctor because I'd have to dissect dead bodies, and that was really gross. And I didn't want any part of it. Yeah, things
But
turned
now you're dissecting brains from dead people.
almost every week. Yeah. Yeah, so it's funny how things go. Your child brain and your adult brain don't always see eye to eye, you know.
Yeah. I, I was going to be a veterinarian. I didn't want to be a medical doctor because I didn't think I could handle like making a bad decision and a patient dying and that kind of seemed very kind of stressful situation. But so then, did you grow up on the East Coast?
I grew up in Ann Arbor, Michigan.
Ah,
Um, I'm very proud to say that this week. Yeah, go blue.
yeah.
Big week for Michigan fans, but yeah, I grew up in Ann Arbor, and my family had moved there when I was under a year old, because my dad was starting a PhD in education, and we
Ah,
stayed throughout my upbringing, and so I was raised in Ann Arbor, which was a wonderful place to grow up.
yeah. Yeah. It's a college town and a lot of energy in the
Yeah,
atmosphere. And then you went to Brown, right, for your undergraduate work?
I did. I graduated from Brown in 1994. Um, and that was another wonderful place to spend time. Larger city, but still a kind of college town, and a very vigorous intellectual climate on campus. And I felt very lucky to be there because of the interactions I had with my fellow students and teachers.
And then how did you end up in San Francisco? You apply for different medical schools and just chose that or
Well, the whole story is actually that I was following a college sweetheart who was from the West Coast, and she wanted to move back there after she graduated. We graduated, and I moved uh, for that reason, and then went to medical school at UCSF as part of that same chapter in life, and then. I started bouncing around a little bit from there on out. I did my residency at Mass General and Brigham and Women's Hospital in neurology after med school, and then came back to San Francisco for my fellowship training in behavioral neurology with Bruce Miller, because by then I had a pretty clear sense that I wanted to study frontotemporal dementia. That was going to be my focus in my career. Didn't know exactly what I was going to do with that, but I I knew that was the disease that interested me. And Bruce was the person to train with if you wanted to focus on FTD. I knew that because he had just arrived at UCSF when I was finishing medical school, and I had my last month of medical school was actually spent with Bruce in a little kind of elective research, intensive month of seeing patients, following him around in clinic, and learning about the disease, which was still very new in the medical consciousness at that time. Really not a part of everyday medical um, you know, uh, conversation. So that was one of the things that was really attractive. It was a big open area, not real crowded, lots of frontier. I don't love tight spaces. I like the openness and the all the unknowns. That was very inviting for me.
And so how does one, as a neurologist, diagnose frontotemporal dementia?
Right. So, I mean, when we say frontotemporal dementia, we're referring to a pretty broad clinical spectrum of illnesses that usually present with a change in behavior or language or movement.
Yeah.
And sometimes it's a combination of all three, and we have different kind of um, recognizable syndromic terms that we apply to sort of describe these particular clinical subtypes, which are each very different from each other and super fascinating individually. And then we kind of house them all under this big term FTD. So when you talk about diagnosing FTD, it becomes useful to be a little specific, and the keys to the diagnosis differ depending upon which kind of specific subtype you're trying to diagnose. That was a long introduction to the answer to your question, but the most common sort of flavor of FTD is what we call the behavioral variant. And this is the one that is characterized by really predominantly social and emotional changes and dysfunction. Um, and so the earliest symptoms are often seen at work or in the family, where there's just a subtle shift in how some person approaches social engagement, interactions, how they, you know, um, tackle their work challenges or their interpersonal, dynamics at work. Something shifts very subtly in there, and you see a sort of flattening of their trajectory because often the symptoms are beginning in midlife uh,
Huh.
among people who are vigorous, thriving members of society, and then that trajectory starts to flatten a little. Um, Promotions are missed. Maybe there's a downsizing at the company, and everybody just assumes that the patient was caught up in that, but really, company had been looking for an opportunity to shift away from that individual because they were just not performing at the same level, and it's not because of cognitive problems, forgetfulness, or disorganization. It's because they can't quite navigate the social and emotional aspects of the workplace in the same way. So when I'm trying to diagnose this particular um, flavor variant, the behavioral variant, I'm listening to the history very carefully for those subtle shifts at home or at work. Um, by the time patients come into the office, oftentimes there's a lot more there. They've developed, um, compulsive or ritualistic behaviors. Their eating habits have changed. They've lost lots of their usual drive toward,
Hmm.
family affairs or personal interests and hobbies. They've lost their ability to regulate social behavior and often do things that are embarrassing to their family members or, might be called tactless or ungraceful socially, commenting on people's appearance or weight, things of that nature are common by the time they come to the office. And so usually when they've gone through years and years of searching and probing and trying to understand what's going on, by the time they come to a subspecialist office like mine, there's the diagnosis isn't so hard. It's just in those earlier years when they were kind of trying to navigate the system from their primary care to a psychiatrist to a social worker, bouncing around from different providers, and then finally someone thinks maybe it's neurology, and then they show up at my door.
And then the variant where there's speech language problems, is that easier to diagnose or not?
Yeah, I mean, those patients tend to get in sooner because when someone develops a primary progressive aphasia, that's the kind of umbrella term we use there. And there are a few different flavors of that one too. Um, when someone develops a primary progressive aphasia, it's very quickly recognized as neurological. No one wonders if it's just a midlife crisis or a
Yeah,
little depression. It's recognized as neurological, so they tend to come a little earlier.
yeah.
And the symptoms kind of fit oftentimes neatly into a kind of picture that tells the story of which parts of the brain are involved, and we can recognize that clinically. And the language variants are also really kind of uh, distinctive and fascinating. In addition to being tragic, patients with what we call a non-fluent variant struggle to get words out. They have speech that is very effortful. They kind of struggle and press and grope and grab for words, just trying to get the words out. Um, Trip over grammar, things of that nature. And then the other major language variant of FTD is called the semantic variant of primary progressive aphasia. And in that one, it's as if the entire sort of leafy tree of knowledge is slowly, the leaves are slowly kind of falling off the tree from the outside in, where only the most, low frequency sorts of words are degraded at first, and it's in subtle ways around the edges of the knowledge sort of structure. And then by the time someone comes to the office, they may no longer know how to draw a zebra or
Uh,
how to describe justice, that kind of thing. Yeah.
and then say someone their career they were a writer, would it be that be picked up on earlier because that's what they do every day?
Absolutely. People who lean heavily on verbal skills will often come in earlier. I had a patient decades ago who was a university professor who was notorious for memorizing his whole student body in terms of their names. He
Uh,
he could call on people by name in a classroom of 300.
wow.
And you can imagine things changed, early. That was a skill that few would have anyway. And then when he started to decline from his illness, that one he imposed a seating chart actually, so we could look up um, the names on a seating chart and start calling up people that way.
Yeah. And then lastly, the motor problem. So kind of the two most common motor disorders we hear about a lot are Parkinson's and ALS. Are the symptoms similar to either of those?
Yes, it's a great setup. So, um, with regard to the motor variants of FTD, there are a couple that we would actually call subtypes of FTD. One is called a corticobasal syndrome, in which patients often in an asymmetric way develop stiffness, um, what we call dystonia, which is an abnormal kind of tone in the limb. Muscle tone is increased. The limb becomes a little bit twisted and difficult to use for skilled movements, in addition to being stiff and sometimes jerky. And that corticobasal syndrome has a cognitive and language kind of component to it also, but it usually starts in a motor domain with
I see.
a hand or a foot that's becoming less and less useful to the patient. Um, Fascinating element of that syndrome in some patients is what we refer to as alien limb phenomena, where the the patient's limb, often it's a hand, but sometimes it's a foot, will do things that they don't intend for it to do. Um, it'll either grasp onto objects or handrails. That limb will levitate sometimes. Sometimes it will even do um, actions that look like they should be purposeful, but the patient swears, I didn't do that. I didn't mean to do that. That's why we refer to it as the alien limb. For example, the better hand might button up a shirt, and then the problematic hand will come and unbutton the shirt. A phenomenon we call undoing. So that sort of intermanual conflict is one feature of the corticobasal syndrome in some patients, and raises lots of interesting questions about agency and how we exert free will over our actions and all kinds of things like that. The other major motor variant of FTD is called the progressive supranuclear palsy syndrome, also known as Richardson syndrome. And this is a falling disease. It begins with gait instability, um, and oculomotor problems, meaning difficulty moving the eyes up and down in particular, which also contributes to the falling. But there's also a very um, consistent and recognizable frontal kind of executive and behavioral disorder that may even come before the motor features and overlaps with some of the features I talked about in the behavioral variant of FTD. So progressive supranuclear palsy also under the FTD umbrella now. And then the final thing related to your question, Mark, is ALS. So ALS, it turns out, is clearly on a spectrum with FTD because many of the patients who present with pure FTD will go on to develop a motor neuron disease of some type. Sometimes it's upper motor neurons only, lower motor neurons only, or both, in which case ALS is the term we would apply. And conversely, patients who come to an ALS specialist often are reporting the recent emergence of some subtle social and behavioral changes or language changes in some cases, a subtle frontal executive dysfunction. So there's a lot of clinical overlap. And then what the last, decade and a half now of research has taught us is that at the genetic and molecular level, the overlap is even stronger. It's really, in some ways, two different presentations of the same disease in some cases, ALS in one patient, even pure ALS, pure FTD in another, same underlying molecular proteinopathy, the same underlying genetics. So tons of overlap there and a really important kind of piece of the puzzle to understand in order to try to make sense of this whole area.
So, but people have heard a lot about Alzheimer's disease. It's generally in the news a lot more because it's more prevalent. And there, the pathology is defined by the accumulation of the amyloid beta peptide and so-called plaques, and then the neurofibrillary tangles. So when you look at their brains, those are and do the right staining and so on, those are the kind of key postmortem diagnostic things. What do you see in the brains of FTD patients after they pass away when you look? And I know it's a little complicated because you got these different variants, but if you could kind of go through.
Yeah, this is one of the richest and most kind of intriguing parts of the whole picture, because in addition to that layer of clinical kind of heterogeneity with all these different syndromes I described, as you could tell, each a little different from each other, there's a comparably nuanced and heterogeneous pathological spectrum that we refer to as frontotemporal lobar degeneration, or FTLD, instead of FTD, that's sitting underneath all those clinical variants. So what do I mean by sitting underneath? Well, what I mean is, you know, depending on how you count, there are about 15 or 20 distinct and recognizable neuropathological diagnoses that we might make at an autopsy that are associated with those, I think, depending upon, again, how you count, 6 to 8 different clinical variants. So, when I try to talk about this to people, I'm often trying to explain, yeah, for each clinical variant, there's sort of a differential diagnosis. We have a little list of usual suspects that we might find at the autopsy, and that list might be 2 or 3, or it might be 10 or 15, and it just depends on the clinical variant. But there are some general things I could say to help distinguish in the minds of your listeners what we see in an autopsy of a FTD case in comparison to an Alzheimer patient. So in a patient with Alzheimer's disease, as you said, there are 2 proteinopathies going on, right? There are 2 protein misfolding disorders. One is the amyloid beta accumulating outside of the neurons in the what we call the neuropil, which is the space in between the neurons that's occupied by all the synapses and the dendritic processes and the glial processes, et cetera. And then those plaques accumulate out there, and then the tangles inside the neurons. In FTLD, we see protein aggregation inside the neurons, and it's usually in the sort of cell body and the dendrites and the axons, the sort of cytoplasm of the cell, as opposed to in the nucleus, but sometimes in the nucleus too. And those protein accumulations are made up of one of a few different proteins, and usually just one. In contrast to Alzheimer's, where there are 2 proteins that are sort of interacting with each other in really fascinating ways, amyloid beta and tau. In FTLD, the aggregating disease proteins are different. So one of them is actually tau again, but a different type of tau, and a tau that's been misfolded in ways that are unlike the misfolding that occurs in Alzheimer's disease. We know all that now because of this incredible revolution in ultrastructural studies that can be done now with cryo-electron microscopy. In the last 5-10 years, we've kind of mapped out exactly how each of these different distinct FTLD tauopathies it misfolds the tau. And they're all a little different from each other and different than Alzheimer's disease, as the pathology would have predicted. But the other one is this really fascinating protein, just as important in FTLD, which is called TDP-43. TDP-43 being a protein that's normally expressed in the nucleus, where it regulates gene expression. It gets mislocalized to the cytoplasm, starts aggregating there, and gets cleared from the nucleus. So it can't do its normal jobs there. And that's a whole fascinating biology that's just really taking off now in the field. And then there's a third least common protein that aggregates in FTLD. It's actually not a one protein, but it's a little family of proteins. And that one's also super interesting. Don't want to get too far into the weeds and derail our conversation around other things. But that family, which is called the FET family, FUS, Ewing sarcoma protein, and TAF15 is a really interesting topic of study, but the least common of the 3. So you've got these 3 major molecules. Each one of those major molecules misfolds in a handful of different ways and produces a different pathological picture. And that's how we get to about 15 or 20 different, distinct diseases that might be the cause of someone's FTD clinical problem. And so it makes it a very rich and fascinating area, but not like something that you can explain in a sentence.
And then there's some cases of FTD that are inherited caused by gene mutation. My understanding, the first genes in which mutations were shown to cause FTD was tau. Is that right?
That is right. Yeah. Um, so the first was tau, and that was a big landmark because it showed that these FTD cases were really different from Alzheimer's, caused by a different biology. Tau mutations don't cause Alzheimer's disease. They cause FTLD with tau inclusions, and the clinical presentation is usually much more of an FTD presentation than something that looks like Alzheimer's. There's some interesting exceptions to that. And then about 10 years later, we got the second of the big three. There's three big genes in FTD. The second one, so the patients who had an inherited form of FTLD with tau inclusions, that first major molecule that I spoke of, those patients have tau mutations in the family. The second major gene we learned about is a gene called progranulin. And in progranulin mutation carrying patients, the molecular pathology is never tau. It's a specific subtype of FTLD with TDP43 aggregation. And so that those two stories actually broke at the same time, just about at the same meeting. I was at the meeting. I'll never forget it. I was a relatively new person to the field, and we learned what the molecular identity of TDP43 was. We knew it was there for years because there were patients who came to autopsy. They had lots of frontal and temporal degeneration, no tau inclusions in the brain, but they had inclusions in neurons that we could see with the microscope using a variety of staining approaches, but none of those approaches told us what the protein was. And then Virginia Lee at University of Pennsylvania, revealed this to the world at this meeting. This is, I guess, 2000 and let's see, 6-ish. And in the same meeting, the discoverers of the progranulin gene told us about their findings that had been previously unveiled, but it was we got to hear both of those discovery stories at the same meeting, so it was very exciting. And the cool thing was that they were linked because the progranulin mutations cause TDP43 to misfold and aggregate for reasons that we still don't understand all these years later. But we've gained a lot of traction in that area, and so that was the second big gene to be revealed. And then in 2011, not so long after, we learned the third of the big three, and that's actually the most common genetic cause of FTD. It also turns out to be the most common genetic cause of ALS, bringing it back to the earlier conversation. And that one is actually a repeat expansion in a gene called C9 open reading frame 72. Boring name. Fascinating disease, fascinating biology, lots to understand there. And this repeat expansion causes all kinds of interesting things, but the usual molecular pathology that we see in patients with that genetic cause of FTD is, again, TDP43. Whether it's a ALS or FTD, either way, TDP43 is the key.
So Brian Traynor discovered that when I was at NIA, and Andy Singleton was lab chief of like the genetics aspect of neurodegenerative disorders, and then I was a lab chief kind of looking at cellular molecular mechanisms, and I remember that too. Now, the progranulin, is that what I had David Rubenstein in one of my podcasts talking about autophagy? Is the progranulin what initially, well, I guess all of these suggest that something's going wrong with the ability of the cells to clear these proteins, but that in particular suggests impaired lysosome function.
That's right. You know, progranulin plays a number of key roles in the lysosome. And if you could see through the wall behind me, there's a colleague
Yeah,
of mine named Amy Gao, whose whole lab is dedicated to try to understand lysosomal biology and progranulin and other forms of FTLD. And so there's a lot of interest in that area. David is another person who's been looking at this across lots of different neurodegenerative diseases, from Huntington's to FTD with tau inclusions and progranulin and others. So yeah, he's kind of a longstanding colleague of mine also, and a guru about all things, you
yeah.
know, autophagy and lysosome.
You know, in our work and others, so in AD anyway, there's a lot of evidence that's accumulated that there's kind of a general, or maybe it's some specific to some brain regions, hyperexcitability of neural circuits that's going on. And AD patients have much more often than age-matched controls develop seizures. And if you do EEG, even if they don't have seizures, they tend to have some evidence of hyperexcitability. And it kind of makes sense to me because the age is a major risk factor for these disorders. And a lot of the things that change even during normal aging tend to kind of make cells prone to hyperexcitability, oxidative stress, impaired mitochondrial function, which can lead to problems in maintaining ionic balances and so on. Is there evidence in FTD that patients early in the course of the disease may have hyperexcitability in the brain regions involved?
Yeah, it's a great question. Yeah, I kind of lived through a lot of the you know, blossoming interest in hyperexcitability in Alzheimer's. You know, my colleagues, Leonard Mucke and Jorge
Yeah,
Palov at
yeah.
Gladstone Institute, sort of discovered that their Alzheimer's disease transgenic mouse models were all seizing.
And
uh,
we, before that, we'd shown that my most highly cited original research article was when we showed that if we expose neurons to levels of aggregating amyloid beta peptide that aren't toxic, they nevertheless make the neurons much more vulnerable to excitotoxicity. So it takes
Yep.
less glutamate to kill the neurons if there's amyloid around.
Makes total sense. Yeah. And, you know, I think that that theme has really blossomed over the past, you know, 20 years in Alzheimer's. In particular, in the FTLD spectrum, we don't have as strong a clinical signal as there was in Alzheimer's disease. You know, the incidence of seizures in patients doesn't seem to be particularly increased. But there are some interesting potential ties to the hyperexcitability idea. And one cool story I'll mention about that. Um, you know, this kind of picks up on the story of the progranulin discovery. After progranulin was identified, you know, the field ran out and does what it always does. It made mouse models of
Yeah.
progranulin knockout, and interestingly, the you know, the patients are all progranulin heterozygotes, meaning they have a usually missense or frameshift mutation that will result in a protein loss of function, degradation of normal progranulin levels. They'll walk around with half normal levels in their, you know, so in circulation. Uh, the mice with a heterozygous knockout, or hemizygous knockout, we call it in mice, uh, don't have a huge phenotype. It's very subtle. Those with a you know, a double homozygous knockout, so complete loss of the gene and protein, uh, do have a phenotype. It's subtle. It comes a little late. It involves a variety of sort of subtle social deficits that my colleague Eric Robeson at University of Alabama, Birmingham has really carefully described, among others. Um, it involves a kind of compulsive grooming phenotype that has the feeling of sort of a circuit overdrive because the mice really they injure themselves by over grooming and scratching at their faces. They take fur off their faces by self-grooming behaviors. And my UCSF colleague Eric Huang, a neuropathologist here, looked at this very carefully in his lab and found that there was a circuit hyperexcitability going on in the mice that was rooted in the thalamus. And the thalamocortical projections were becoming disinhibited due to a very specific loss of inhibitory input from the thalamic reticular nucleus neurons onto the thalamocortical cells. And that specific loss of inhibitory synapses seemed to be causing this thalamocortical circuit hyperexcitability. And right around that same time, I was working with one of my colleagues here, Susie Lee, on a study where we were trying to look at the patients with progranulin-related FTD, but even more importantly, their asymptomatic relatives who carried the gene but didn't have any symptoms yet. And what we found in those asymptomatic relatives who are in this sort of pre-symptomatic or prodromal stage of FTD is that their thalamocortical network activity was intensified compared to controls or even family members without the mutation.
And
And
that's by fMRI?
using fMRI, yeah, looking at network kind of cohesiveness, what we found was a very strong signal pointing at the thalamus in a bunch of its different thalamocortical interactions showed this sort of
Interesting.
hypersynchrony going on, detectable even with a tool like fMRI. So it's very, very interesting kind of convergence between what Eric Huang and his lab were seeing in the mice and what Susie Lee and her group and collaboration with my group were sort of seeing in humans. It just all kind of fit together at the same time. And we're still interested in that. And there are even companies now that have cropped up to try to do focal targeted, gene therapy by injecting into the thalamus. So it's become a fascinating area to be part of in that way.
So
And so,
there's companies that they plan on doing clinical trials. Is that,
yes, clinical trials for progranulin FTD are already underway.
huh?
And some of those are based on specific kind of mechanistic targets that can be approached with antibody mediated therapies and things like that. But others are actually gene therapy trying to
Okay.
go and restore progranulin levels in people who have granulin
Wow,
related.
interesting. And they're using like adeno-associated virus or,
Exactly. That's the idea with the thalamic injections is an
yeah.
AAV, injected into the thalamus to try to distribute, the transgene out into the or the genetic repair into the
But
into
these are the kinds of things we've done in animal models for years, and now they're, well, that's really exciting. Let's hope it helps.
yeah, no, I think that's right.
Okay, so looking back in your publications, you were interested in, and I mentioned this earlier, in selective neuronal vulnerability, early in your career, and one of your early articles I saw was on these von Economo neurons. You know, I guess you're trying to, is there some type of neuron that's in humans, but not, and I should say in mice and rats, they don't normally develop any of these pathologies as they age, whether it's Alzheimer's like pathology or FTD or ALS. So we don't know why. It could be a lot of explanations. But anyway, so could you briefly talk about that and what your current thinking is on why some neurons are particularly vulnerable?
Right. So I mean, this is a theme that
That
cuts
is,
across all of the degenerative diseases. You know, each of them has a stage during which the major pathological event of protein misfolding and aggregation is occurring within a very circumscribed region or regions, and only a few cell types, or even just one probably in some cases. And the example that makes it easiest to talk about this is probably ALS. We know it starts in the upper and lower motor neurons, sometimes one, sometimes the other, variable combinations of those two um, sets of deficits in the patients. But that same principle that ALS illustrates so beautifully applies across all the neurodegenerative diseases. And um, when I entered the field in the late 90s, and was reading about FTD, what we knew about its anatomy, um, I was just really struck by how little we knew. Um, you know, uh, Despite some of the pioneering work that was done in brains by a guy named Arne Brunn, working in Sweden, um, we still had a very kind of rudimentary understanding of where in the brain FTD really went at the very beginning. We refer to it as frontotemporal, but as you know, Mark, frontotemporal is a lot of real estate.
yeah.
That is more than half of the brain. So it's a lot of real estate, and I was really struck by that and curious about it. But at the time, being a relatively junior researcher with no particular training in neuropathology, neuroanatomy, just what I got as a neurology resident, um, I I didn't have much in terms of a skill set to go after any of this. But for whatever reasons, I'll never understand entirely. That didn't stop me. So
But you oh, this was before you were at UCSF, or
no, this is just after I
oh,
got there.
there's some good neuropathologist, Steve Darmond, right?
Yeah, he's a big part of the story. So
Oh, okay.
yeah, so I guess I should clarify that my training was all in neurology. Never trained as a neuropathologist.
Uh,
And and so when I got to UCSF to work with Bruce Miller, my mindset was that I was going to do clinical research on FTD, try to learn from the fascinating symptoms, and maybe learn something about the anatomy, use some brain imaging techniques, something like that. That was a little bit kind of in my comfort zone, my prior training path, and I did that. But along the way, um, I got interested in this population of neurons that you referred to called von Economo neurons. And this was very serendipitous because my only awareness of them came from a lecture that I heard as a fellow at UCSF. It turned out Steve DeArmond, who you just mentioned, had invited a guy named John Ullman, who is an evolutionary biologist and really legendary vision researcher at Caltech, a really amazing guy, and he's still working to this day. He came to UCSF. He spoke about these neurons that were localized. This was the key piece to these structures that we had just been learning were the earliest targets in FTD, the anterior cingulate cortex and the anterior parts, especially the ventral anterior parts of the insular cortex. And when he presented that information, it just hit me over the head, like a ton of bricks, because here we were in FTD, we have no sense of the early vulnerable neuron. And then there's a neuron that's uniquely specialized for social functioning in humans and great apes. It turns out other large-brained mammals as well. And it's localized to the two areas that are the earliest affected in FTD. All this kind of came together at once. And so that created this huge shift in my career plans. And it resulted in me seeking out Steve DeArmond as a mentor to teach me neuropathology because I hadn't been trained in that. And Steve is an amazing neuropathologist,
yeah.
one of the best. And so I got to work in his
And
lab.
he did all the neuropathology for Stan Prusner and
Exactly.
that
Yeah,
stuff.
he
Yeah,
he was the neuropathologist who pioneered our understanding of how prion diseases affect the brain
yeah,
pathologically. Yeah.
yeah, yeah.
And so he had that whole kind of experience and set of, life lessons in his mind when he,
Yeah.
welcomed me into his lab to study FTD and to look at, this question, this hypothesis of whether FTD might target those von Economo neurons in the earliest stages. And working with Steve and John Allman and others on that project was a huge tidal shift in my career trajectory because it meant that all of a sudden I could take my questions to the cellular and molecular level, and that's where I've stayed for a long time.
Yeah.
And you know, it just it's always fun to think back on that stage because I think back on it with a lot of gratitude toward the people, including Bruce Miller, who had the sort of um, uh, courage to sort of support me in going in a whole new direction that I hadn't anticipated at all. And it's turned out to be a sort of guiding light in our laboratory. That's what we still work on, this question of why do these von Economo neurons seem to be the early target in FTD. Now, when I say that, I should be specific because I don't think that they're targeted at the beginning of primary progressive aphasias or movement disorders. That wouldn't make any sense. They're localized in the social and emotional parts of the
Right.
brain, and so it's that behavioral variant I spoke of that seems to target these cells early, and that's what we're trying to understand because it's the one lever we have in the FTD space to look at these questions of cell type specific targeting. In the other variants that I spoke of, we really don't know what the early cell type might be. I think in the era we're living in now, we're going to get information about that soon because of all the amazing techniques that are becoming available.
Are those
Uh,
the von Economo neurons, are they glutamatergic? They use glutamate as a transmitter? Or?
yeah, just real briefly and without getting too deep, but the von Economo neurons are very, very large layer 5 glutamatergic projection neurons that send axons all the way to the brainstem, if not beyond, definitely to the brainstem, where they, synapse onto the major autonomic integration sites like the nucleus of the solitary tract, um, parabrachial nucleus, PAG, and others.
So do these patients have autonomic dysfunction?
Well, we never knew that, but when we learned about these von Economo neurons and their place within this larger network that is targeted in FTD, we went out and studied it, and it turns out they do. Um, Patients with BV-FTD, when they're just sitting quietly in a laboratory, have lower sympathetic and parasympathetic tone compared to
Huh.
healthy individuals. They have a degraded baseline autonomic milieu, is one way to put it. And when they're stimulated by an emotionally powerful stimulus, like a film clip or an awkward interpersonal situation or something like that, they have a lesser response of the autonomic nervous system to those stimuli as well.
Huh.
And we had always been thinking, oh, well, they're blunted, but it's just because they don't understand the stimulus or they don't care about the stimulus, and all of that might be true too. But in addition, they have, a primary deficit in autonomic tone.
And do
Uh,
they have any, is their exercise tolerance normal? Is their
right.
cardiovascular regulation normal?
Great question. When you push it that far, it seems to hang in there okay.
Oh, okay.
It's just these subtle things where, we use these subtle emotional and autonomic responses to guide so much of our behavior. And those subtle cues and signals that we get from our bodies are what I think is kind of degraded, even in the earliest stages of the
Yes.
illness. And so it's a fascinating kind of set of things to think about where, maybe it doesn't take that much to set someone off course behaviorally, but a sort of blunting or degradation of these subtle autonomic cues. I mean, it's a fascinating thing to think about.
And in Parkinson's, one of the early clues that there may be some problem with the parasympathetic nervous system was they have chronic constipation, right? And
Yeah.
now I had Ted Dawson and others on to talk about this Brock's staging and all that. The disease may actually start in the gut and go up the vagus to the brain. Is there any evidence for like the pathology actually? Has someone looked in the enteric neurons, the neurons that innervate the gut in FTD?
For the most part, it remains a bit understudied. We don't have great reasons to think that um, there's a peripheral onset to FTD like there is for Parkinson's.
Okay.
Um, and for the most part, that hasn't been studied. We put out one case report that you might find interesting in this regard. We had a patient with um, what was a kind of atypical variant of behavioral FTD. Uh, but one of the things that made her atypical, among others, is that she had really severe abdominal symptoms. She was in and out of emergency rooms all the time for abdominal distress, vomiting, diarrhea, etc. Studied like crazy by gastroenterologists, never found anything to be wrong. And it turned out that she did have FTD, and she even had one of those C9orf72 expansions that I mentioned earlier. And in her case, because she was so atypical, she underwent a whole body autopsy. And so we were able to go and look at the enteric nervous system. And sure enough, the C9orf72 related pathology had been in kind of affecting her neurons in the gut and altering the way those were working and resulting in all those symptoms. So super fascinating, probably more of a one-off than a regularity though, because we usually don't hear those symptoms. Usually we hear that a much more prominent
But I
picture.
guess I
But
guess in that case, when you have the mutation, it's in presumably in all neurons,
every cell of the body, sure,
right?
yeah.
So you can imagine that a lot of neurons are going to be affected in that.
Yes, yes. And despite that being the case for all the patients with that expansion, we still never hear these GI symptoms. I think she really was atypical, but
Okay.
it showed us things that the disease can do, not necessarily what it does all the time. Yeah.
Okay. Last two things, Bill. Uh, First, there's a lot of interest in these age-related neurodegenerative disorders and kind of identifying patients even before you can see the clinical symptoms. And so biomarker development is a lot of activity in that area. And in fact, the neurologist that was under me at NIA, Dimitros Kapagiannis, is doing a lot of work with these, they're called exosomes or extracellular vesicles. And he uses antibodies that are specific for neurons to take blood samples and pull these small vesicles out that he thinks are coming from neurons. I guess can't be sure exactly where, but and then kind of interrogate them. And he's seeing some nice work even in longitudinal studies and where he has blood samples from people before they developed Alzheimer's, and he's seeing elevated uh, tau, phospho-tau or whatever in there. Is there anything like that going on with FTD?
Yeah, there is, and it's really emerged as one of the major research priorities for the field. Um, you mentioned pre-symptomatic detection, and that would be amazing.
Yeah.
But in FTLD, we have an even bigger problem when it comes to conducting clinical trials right now. And all I have to do to explain it is to remind you what I said earlier. We have
There you go.
15 different
Yeah.
pathological diagnoses, three major molecular classes. And when a patient comes to my office with behavioral variant, I can't really say which of those three classes they belong to. And so I have no way to sort them into the right trial.
Yeah.
So that has huge implications for the field. And what I hope will happen over the next 5 years is that there'll be a whole lot more energy and investment directed at biomarker discovery for molecular diagnosis in FTLD. I think this is critical as we move toward trying to find therapies. It's been a huge key to unlock therapies in Alzheimer's disease. Having the availability of those molecular biomarkers
Yeah.
has made all the difference in my view. And we need that for FTLD for reasons that are even more fundamental. Getting the diagnosis right, sorting patients into the right kind of trial, and then, of course, monitoring the results of therapy or detecting it pre-symptomatically would be additional, goals for research. And I think there is room for hope in that area. I think the tools are maturing, the investment is increasing. A lot of pharmaceutical companies want to invest in the area of FTD drug development now, which is amazing. But they also are a little wary about the lack of biomarkers, so they are also investing in this area, which is great. Can't wait to see what comes of all that energy. I think the key is going to be, going after the specific misfolded proteins or certain specific consequences of that misfolding. And yeah, the tools are really getting ready for this.
And so you mentioned the one clinical trial of a gene therapy for the progranulin loss of function mutation patients. What else is going on? One thing before I retired from NIA that we got involved in fairly heavily, and it's continuing, is that has to do with like bolstering bioenergetics in neurons with ketone esters, for example. There's clinical trials going on now in AD with that, and that's based on, as you know, one early thing that was found functionally in Alzheimer's was impaired glucose utilization using radio-labeled glucose and then PET imaging. And Steve Cunan up in Montreal, he's shown that um, he has radio-labeled ketone, acetoacetate, and does PET simultaneously with FTG, fluorodeoxyglucose PET. And so he's found that when people are in a ketogenic state, their neurons switch to using ketones, just in healthy people. And then he has evidence that at least in early Alzheimer's disease, neurons can still use the ketones. And anyway, if I was going to propose a trial in FTD, that's right, because it's very safe and something that's worth a shot. And if it worked out, it could be something relatively inexpensive. Anyway, what's going on? There's a lot of different approaches. Stimulation. I had Li Wei Sai talk about this gamma frequency stimulation for Alzheimer's, and then I don't know how much time you have. We can take a few more minutes, I hope.
Yeah, sure, no problem.
Okay. And then, other forms of stimulation, either with implanted electrodes or transcranial magnetic stimulation. So I guess kind of to finish off, can you kind of give a flavor of what's going on? And
Overview of the
yeah,
experimental therapeutics landscape kind of thing. Yeah.
yeah.
Yeah. I mean, I have this feeling that the experience we'll have in the FTD field will be a bit different from the Alzheimer field for the simple reason that um, setting up and powering a trial in FTD can be pretty challenging.
Hmm.
Uh, and so I feel like a lot of the early shots on goal are going to be very molecularly oriented, strong proof of concept in place, often focused on the genetic forms of the disease because they um, sort of provide a workaround for the problem of molecular diagnosis that I spoke of a moment ago. But because, you know, FTD is only caused by a known gene in about 20 to 30% of all patients, and that share is divided up three ways among these big three genes that I spoke of. powering the genetic trials is difficult because of the small number of patients worldwide available, and then organizing a trial for sporadic FTD with no known gene is difficult because of a lack of clear molecular diagnosis. So the landscape is a little influenced by some of these contextual factors.
Yeah.
And so right now, a lot of the trials under development are focused on those genetic
Yeah.
forms where we know exactly what the molecular pathology will be. And they are, targeting patients who are already symptomatic at this stage, but I think we'll see a shift, hopefully sooner than later, toward going even earlier in the pre-symptomatic stages, because we know how to define those and identify those subjects. And a lot of researchers, including those in my group, are working on trying to develop early detection methods for when a carrier actually starts to convert over. Very important question for organizing trials. But a lot of the energy is in that genetic FTD space right now. What I hope will happen as companies and other and academic, treatment-oriented researchers get into this space more and more. What I hope will happen is that we'll have a lot more shots on goal that are a little bit more general in terms of, physiological mechanisms like the ones you were talking about, as opposed to pathological mechanisms, symptomatic relief. We just have had so few trials in FTD in any of those areas because of some of these contextual factors. But I think we're going to see more and more in the coming decade of trials of all these different sorts, not just those directed at the molecular pathology, but for the sporadic disease, for the patient who has no known gene and uncertain molecular pathology, I think the key to unlocking trials is that biomarker piece we spoke about, getting the
Yeah.
molecular diagnosis clear.
Yeah.
There are some of the FTD syndromes, I mentioned this earlier, I'll come back to it briefly, that have a shorter pathological differential diagnosis than others. So for example, uh, about 85+% of the patients with that semantic variant of primary progressive aphasia, the one with the leaves falling off the knowledge tree, about 85% of those have FTLD with TDP43 inclusions. So you could gather a cohort of patients and be fairly confident about the underlying pathology. The same is true for that falling disease, progressive subnuclear palsy. Most of those patients have a tau-based disorder. You could be fairly confident there. And that's where we've seen the most investment by, pharmaceutical companies
Yeah.
is in those few of the FTD syndromes where the molecular diagnosis can be made with confidence during life. So I hope those cohorts will serve as a good, testing ground for some of these general mechanisms, physiological and symptomatic treatments. I think we're going to see more and more of that. Here at UCSF, one of my colleagues, Peter Ubenkov, has just started a trial on that semantic variant, looking at, the inflammatory pathway, neuroinflammation as a potential treatment target. Excited to see where that goes. It's the first trial of its kind for sporadic FTD. So, it's just the beginning right
Yeah.
now. It's just starting. But that's a nice place to be mid-career.
Yeah.
I've gone from, starting as a fellow. We don't know much about it, the biology. We don't know much about the anatomy. We learn all this we have this incredible learning curve over the coming 20 years to where here at mid-career for me, we're starting to see real trials that have plausible mechanisms. And, my great hope is that we'll succeed in finding treatments for these patients during my career. I mean, that is my great hope. And I do have that expectation. I have to say, and I couldn't have said that when I started,
Yeah.
but I do have that expectation now. And the success in the Alzheimer field has, buoyed everybody's hopes about this. It can be done, and we've seen the role that biomarkers can play and the impact they can have on this. So I think there's a lot of optimism um, in the field. The targets that we've been hitting in Alzheimer's are the same ones that were identified through neuropathology in the original description of Alzheimer's.
Yeah,
You know, it took a long time and a lot of work and a lot of refinement in the tools to get to the point where we could hit those targets and make a difference for patients. FTLD is on an accelerated time scale. You know, we're working in an era where the tools are more sophisticated and mature, and it
yeah,
can all move faster, in part because of the you know the hard, long, you know, continuous effort that went into the Alzheimer field and laid out a pathway for this.
yeah.
And so it's a wonderful place to be here now for me mid-career to say optimistically, I think we're going to have treatments for FTD during my career.
Okay, that's a great place to end on a positive note. And thanks for your time, Bill. I know you're busy. I'm not so busy since I retired.
Well, you're making
And
good use of your time, Mark. It was a pleasure to talk to you. Thank you for inviting me. I really
yeah,
appreciate the
yeah, I
conversation.
enjoyed. I don't recall if I'd met you before or not.
I don't think so. I've
I
been,
was at University of Kentucky for 11 years, and Bill Marksberry was. I'm far from being a neuropathologist, but I learned a little bit from him about neuropathology.
yeah, he's a legend for
Yeah,
sure. Yeah.
yeah.
Um, yeah, no, I don't think we had met before, but been aware of your work for a long time and really appreciate the chance to talk to you. It was really fun.
Okay, have a good year, and I'll let you know when comes up.
Sounds good.
All
All right. Take
right.
care.
Yeah.
Bye-bye.
Bye.